Good afternoon. I'm Federica Piccirilli. I currently work at the CC beamline at Elettra Synchrotron with the group of Lisa Vaccari. I've been working in infrared spectroscopy for 10 years, and I joined last July the RIT Institute of Area Science Park in the framework of the PRP at CHERIC project. In the project, I'm involved in all the activities related to the development and the installation of a micro to Nanoresolved and station working in the infrared spectral range. In this seminar, we will be talking about the Nanoresolved infrared spectroscopies applied to the study of biological matter. I thought it was a good idea to divide this presentation into two main parts. In the first part, I would like to recall some basics on infrared spectroscopy. I will tell you something about the merit and the limit of transmission infrared microscopy, the diffraction limit, and we will talk also about the near-field approach exploited in the last 10 years with a specific aim to push the lateral resolution of infrared microscopy to the nanoscale. And we will see two different techniques, the s-SNOM and the PTE. In the second part, that is the part on which I would like to focus more, we will see practical examples with experiments from literature and experiments we performed at the beamline, exploiting one of the two nanoscopies that have been developed up to now, that is the s-SNOM. If we get time at the end, I would also like to tell you something about another technique that is not properly nanoscopy. It's a sub-micron technique allowing to overcross the diffraction limit imposed to the infrared spectroscopy, and that is also relevant in the framework of the project. So starting back to the start, infrared spectroscopy is the technique exploiting that region of the electromagnetic radiation that is in between the visible light and the microwaves. In particular, in the region between 1000 centimeter minus 1 and 4000 centimeter minus 1, the so-called region of mid-infrared spectroscopy, all the vibrations of covalent bonds falls, and it is possible to, by performing studies on this spectral region, to obtain the fingerprint of organic compounds. Starting from a very basic example, an isolated water molecule, if we acquire the absorption spectrum of water, we will see in the mid-infrared range the presence of three main peaks. These peaks are related to the vibrations of the OH covalent bonds and can be easily calculated. Whose frequency can be easily calculated considering water molecule, considering the covalent bonds in water molecule as harmonic oscillators. If we do a parallelism, direct parallelism with a much more complex system as an eukaryotic cell could be, we can see that there are a plethora of peaks in this same spectral region that can be associated to the bonds belonging to different cellular subcomponents. So it is possible from the observation of the spectral position and the relative intensities of these peaks to obtain important information on DNA conformations, protein secondary structure, and lipid phases. The infrared spectroscopy comes together with the infrared microscope, allowing to perform spatially resolved infrared studies that has the limit for the lateral resolution that can be achieved that is imposed by diffraction effects that cannot be neglected when the size of the object that one wants to probe is comparable to the wavelength of the light that you used to probe it. To give you some numbers, the diffraction limit at 1000 centimeter minus 1 is of 10 microns. This means that if we want to probe the spectral characteristics around this wave number, we can do it with a pixel size that is at this minimum size of 10 microns. De facto, this lateral resolution can be achieved only with the synchrotron source, that is a highly brilliant source delivering light in a very small surface in the range of tens microns. And to give you an idea of the studies that can be performed with micro FTIR, it works quite well with single cells, so it is possible to acquire single cell spectra. Here there is an example of a study performed with synchrotron light on cells, and here in this panel it is shown the spectrum of in the region of proteins, showing differences observed in samples treated with specific drugs. And another example of application of micro FTIR spectroscopy is the study of tissues, in which it is possible to get, mainly through statistical analysis. Here, a principal component analysis is, for example, used. It is possible to depict the chemistry of the tissue with the resolution of tens of microns. So given the high sensitivity of the technique to the chemistry of the system, it was highly desirable to overcross the diffraction limit imposed to far-field spectroscopy. And for this reason, in more or less 10 years ago, infrared nanoscopies have been developed. These techniques are based on the coupling of the atomic force microscope to infrared sources and allow to obtain a lateral resolution that is comparable with the size of the atomic force microscope tip, so around tens of nanometers. There are two different approaches that can be used to perform infrared nanoscopy. The first one is a scattering-based approach, in which the probe is optical, and is s-SNOM, standing for scattering type scanning near field optical microscopy. And the other one is a photothermal expansion microscopy. I don't want to go into the details of these techniques. I just want to mention here that s-SNOM is a suitable technique for to be coupled with a synchrotron light, and allows for a penetration a study of a sample surface with a penetration depth that can be tuned depending on the setting of the measurements between 10 and 100 nanometers. It allows the in-depth profiling of the sample surface, and the spectrum that is acquired is polarized along the tip axis direction. This means in practice that all the samples having a strong alignment of covalent bonds can be good samples for the s-SNOM, because it is possible to obtain information on further information on the ordering of molecular structure through this specific aspect of the technique. About the photothermal expansion version of the infrared, nano-infrared, here the spectrum is comparable with the one acquired with far-field spectroscopy. The measurement is more bulky, so it allows to probe a sample thickness of 100 nanometers or more, depending on the sample characteristics, and it works quite well with the samples having a high thermal expansion. And this is because far fields here are probed thanks to a thermal effect occurring on the sample that expand under infrared irradiation. In the next slides, we will focus more on s-SNOM spectroscopy, because it's a technique that we used more at the beamline in the last years, and also because this technique will be exploited in the framework of the PRP at the CHERIC project, with the installation of an instrument allowing to cover the full mid-infrared spectral range. So in s-SNOM, just to give you very few details, the light is squeezed with the AFM tip acting as an infrared antenna, and it generates a dipole-dipole mirror interaction between tip and sample, and the tip is tapping onto the sample, and through the analysis of the backscattered infrared light, it is possible to obtain the optical information of the sample surface up to a thickness that is between 10 and 100 nanometers, as we saw before. Going to more practical examples, I would like to start with a paper from literature that is that kind of papers that everybody would have liked to publish. Here we have the comparison of an electron microscopy image of a section of a cell. The correspondent AFM map, topography map of the cell, and in the colored spot, the authors acquired the spectra that are reported in the panel in the right. Here it is possible to spot the presence, for example, at point 2, 3, and 4 of amide-1 and amide-2 bands that are specific for proteins. And for example, at point 1 and from 5 to 9, the peak of carbonyl groups. So the authors in this paper were able to associate to a specific topography of the cell the presence of its subcomponents. So if you are curious, I strongly suggest you to refer to this article about s-SNOM spectroscopy, because the authors did much more than what I'm showing here. So moving to another experiment, I wanted to show you this example because here we studied a very thin layer of DNA molecules adsorbed onto allocyte fibers. Allocyte is a clay, a natural clay. It is biocompatible and is a candidate for drug delivery and gene transfer. What we wanted to understand here is what are the structural modifications that DNA need to undergo in order to make this interaction possible, since the surface of these two compounds, the surfaces of the two compounds are both negatively charged. So we did an AFM analysis with the higher s-SNOM sensitivity, and we compared the analysis also with scanning electron microscopy. We compared the spectra acquired on DNA, on pristine DNA at far field and at near field, so with conventional FTIR and with nanoresolved FTIR. We compared these two spectra to the spectra of pristine nanotubes and nanotubes loaded with DNA. So first thing that can be easily noted is that while the spectrum of the nanotube is flat, in presence of DNA, it is possible to spot the presence of several peaks that has a direct correspondence to the peaks of the DNA molecule. From the observation of the intensity of specific peaks, it was possible here to understand that the possible opening of the double strand of DNA occurs in order to make this interaction favorable. A similar system similar to the one I just shown, that is part of the PhD project of Martina Zangari, is the case of a very simple model for asbestos bodies that are formed in lung and caused by asbestos exposure. Martina addressed the very debated topic of the protein misfolding induced by these fibers by studying specifically the effect of asbestos fibers to iron-containing proteins, and she focused on ferritin. So here there is the electron microscopy analysis performed on an asbestos fiber, a chrysotile fiber loaded with holoferritin, so with the protein containing a iron core, and the adduct formed with the protein with the empty core, and she compared these two samples with the pristine chrysotile, finding that there is an increasing in the roughness of the surface of the fibers when the protein is interacts with them. So in order to understand the chemical nature of the skin that was distributed around these fibers, we performed an s-SNOM analysis, and here it is possible to see the AFM topography of the adduct formed with holoferritin, and what we did here was with the spectroscopic probe looking for the specific bands of the protein, and we found out that both amide-1 and amide-2 bands are visible. This is a first suggestion, because since the two bands are visible, and being amide-1 associated to CO stretching and amide-2 associated to CN stretching, it means that we are probably dealing with an isotropic system with no specific alignment of bonds. Second, we acquired the imaging map at single frequency by looking at the intensity at the peak of amide-1. What we found by taking the signal coming from different thicknesses on the surface of the sample is that when the protein loaded onto the fiber is an iron core full, the distribution of the protein is more volumetric than when in the case of the apoform of the protein, forming instead a skin around the fiber. And this can be possibly associated to the modulation of the iron core of the distribution of charges on the protein surface. An experiment I would like to show you, because this is the case of aligned bonds in the system, is the case of collagen fibers. In these measurements, we wanted to the main aim was to trying to understand if differences in the secondary structure of the fiber occur depending on its morphology. We are still working on the analysis of this data, so I will show you what we get up to now. This is the topography of a collagen fiber. So these two are the AFM maps, the Z map and the phase map. And here there's the optical amplitude map that is similar to a reflection image. Here it is possible to spot the periodicity of the D-band of DNA in both morphology and optical amplitude maps. And this is a direct proof of the confinement of light to the very nanoscale. Going to the spectroscopy, we observed that comparing the spectra acquired on collagen to the spectra typically acquired for an isotropic system, there is damping of the amide-2 intensity that is due to the radial symmetry of the system, so to the high symmetry of CN bonds that lay on the sample on a surface that is perpendicular to the s-SNOM tip axis. Another example I want to show you here, where the thickness of the sample is at the very limit of the technique. We had to deal with the amyloid-like fibers for self-assembling tripeptides, having a thickness of around 10 nanometers. And for these experiments, there were two things complicating the analysis. The first one is the thickness that is at the limit of detection. And the second one is the fact that amyloid fibers has a very strong alignment of CO bonds being formed by a cross beta structure. So in this case, the amide-1 bonds were on the plane perpendicular to the fiber axis. For these measurements, for these experiments, we had to perform a statistical analysis. We had to go for principal component analysis in order to understand what is the detail of the chemistry, allowing for the activation on one tripeptide having a specific chirality of particular properties like the synthesis of gold nanoparticles. Last experiment I want to show you was on the receptor binding domain of SARS-CoV-2. We studied the effect on the structure of the protein when interacting with the alumina nanorods proposed as vaccine adjuvants. The analysis we did here was on the evaluation of qualitative evaluation of the secondary structure through the convolution of the amide-1 band in its main components. This deconvolution has been performed through the observation of the second derivative analysis, and we found out in this case that first the system is fully isotropic because the conventional the spectrum obtained through conventional spectroscopy is very similar to the spectrum obtained at the nanoscale. So there are no polarization effects, and we also got some information about the concentration ratio for RBD and alumina nanorods that maintains the structural properties of the protein in its native state, finding out that when the ratio is 3:1 for RBD to alumina nanorods, there is a certain fraction of proteins undergoing misfolding. So just a few words I would like to spend on another technique that is the FTIR, as I promised. As I said at the beginning, this technique is not properly a nanoscopic technique. It doesn't allow to go to the high resolution of s-SNOM, but since here a visible probe is used, and the technique exploits the thermal expansion of a sample under infrared irradiation, the diffraction limit is shifted to the visible range. So this is a submicron technique, and offers the advantage to for a simultaneous acquisition of the Raman spectrum of the infrared spectrum and fluorescence. And this technique that is very useful for the characterization of tissues and cells. This is the extended infrared family working at Elettrasynchrotron that I would like to thank because we have a continuous sharing of science and ideas with them. Just a take-home message I would like to leave you. The power of nano-IR resides mainly on the fact that topography with a s-SNOM or with also PTE measurement can be associated directly to chemical features, and this is very important when going to the nanoscale. There are several approaches that can be used in order to achieve submicron to nanoscale resolution, and it is also fundamental. It is fundamental to find the right technique depending on the characteristic of the sample that one wants to measure. The micro to nano end station is currently operating at CC beamline at Elettra, and it will be further extended during this year with the installation of new instrumentation from IRAS and Spark in the framework of the project. And of course, collaborations are more than welcome. Grazie.